From the University of Wisconsin-Milwaukee's Division of Research, welcome to Research Matters. Welcome back to Research Matters, where we spotlight standout research happening across campus and beyond. Today we're diving into a paper from UWM's Department of Physics, along with outside collaborators. A study that doesn't just answer a biological question, but also pushes the boundaries of structural methods themselves.
The team, led by Marius Schmidt and his colleagues, used single-particle cryo-electron microscopy, or Cryo-EM, to visualize a bacterial light-sensing protein in unprecedented detail. And before we go further, let's pause for a quick explainer, because this technique is central to the story.
Cryo-EM is a structural biology method where scientists rapidly freeze proteins so quickly that water turns into a glass-like state, not ice crystals, preserving molecules exactly as they are in solution. Then, using a high-powered electron microscope, scientists collect thousands or even millions of two-dimensional images of individual protein particles. Advanced computational tools align and combine those images to reconstruct a three-dimensional structure at near atomic resolution. In other words, it's like taking a massive number of snapshots of tiny molecules, each at a slightly different angle, and using software to assemble them into a detailed 3D model. This technique is especially powerful for flexible, dynamic, or large proteins that are hard to crystallize. And that's exactly why it was chosen in this study.
So what did Schmidt's team accomplish with Cryo-EM that hadn't been done before? They captured the structure of a wild-type bacterial phytochrome, a kind of light-sensing protein, with its natural histidine kinase domain still attached. That's the enzyme that actually performs the downstream signaling. Until now, structural studies of these proteins lacked that entire region. That gap has been a big obstacle. Without the kinase domain, we could see how the protein absorbs light, but not how that light actually triggers the cell's signaling machinery. Cryo-EM made it possible to visualize the whole system, sensor plus output, for the first time.
And it gets better. The team captured the protein in 3 different functional states, including a never-before-seen mixed or heterodimeric state, where the 2 halves of the protein are in different conformations. This asymmetric state was thought to exist, but this is the first time anyone has captured it directly in an unmodified bacterial phytochrome.
Here's where the implications widen. Many bacteria use what's called a 2-component signaling system, basically a sensor that detects an environmental cue and a kinase that passes the message along. These systems are everywhere in bacterial physiology, from stress response to development.
By showing how to visualize the entire signaling complex and its intermediate states, this paper provides a general roadmap for studying other signaling proteins that were previously out of reach. Cryo-EM lets us capture molecules that are dynamic, asymmetric, and flexible. That's a big leap forward for structural biology.
It also deepens our understanding of how conformational changes travel long distances through proteins. In this case, the team found that subtle kinks that appear in the protein's linker helices help transmit the light signal over nearly 100 angstroms from the light-sensing domain to the kinase. These kinked intermediates may be a common feature in sensory systems.
The authors point out that this work sets the stage for time-resolved Cryo-EM, where you can freeze proteins at different moments during a reaction. Imagine having a molecular-scale, slow-motion replay of a signaling system turning on. That's the frontier this paper pushes us toward.
And for researchers in other fields, synthetic biology, microbiology, bioengineering, computational modeling, the idea that we can now capture full-length, functional protein complexes in action is a huge opportunity. Whether you want to design light-controlled pathways, understand bacterial development, or explore new antibiotic targets, this method opens the door.
If you're curious to see how this breakthrough was achieved, check out the full open access article titled Photoreception and Signaling in Bacterial Phytochrome, revealed by single particle Cryo-EM, published in Science Advances in August 2024.
Take a read, share it with students, or spark a cross-department collaboration. Because this kind of methodological innovation doesn't stay confined to one discipline.
That's it for today's episode of Research Matters.
Until next time, stay curious.
Research Matters is produced by UWM's Division of Research, which reserves the rights to this audio podcast. All other copyrights are held by the publication authors and produced with their permission and cooperation. For more information on UWM's Research Podcast, please visit www.uwm.edu/researchpodcast.